Welcome to our today's lesson. Today we are going to do a practical on preparation of a slide. That is a temporary slide. Now, when you're using a microscope, we need a slide. So we have two types of slide. We have a temporary slide and we have a permanent slide. A temporary slide is prepared in the lab for immediate use, while a permanent slide is prepared and preserved for future use. But today we are going to prepare a temporary slide. For us to prepare a temporary slide, there are things that we require. Number one, we are going to require a microscope, it is. Number two, we are going to require distilled water here inside the wash bottle. We are going to require a dropper. We are going to require iodine solution. We are going to require a pair of forceps. Also, we are going to require a mounting needle. Also, importantly, we will require clean slides, clean cover slips, and a scalpel. To use. Apart from these apparatus, we also require a specimen. We can use an onion bulb, or we can use a seedling from a bean or a maize. When using a seedling, we cut thin section so that light will pass through. So what are the procedures of making a temporary slide? Procedure number one, you have to cut the bulb onion into 4 parts. So I'll cut it into 4 parts. So procedure number one, you cut the bulb onion into 4 parts. I've cut mine into 4 parts. Procedure number two, you separate a fresh leaf from one part. So you separate a fleshy leaf from one part. So here is our fleshy leaf from one part. Procedure 3, we are going to peel a piece of the epidermis from the leaf. We are going to peel a piece of the epidermis from the leaf, and we are going to use a pair of forceps. So I'll demonstrate. So here is my epidermis of the bulb onion. We go to procedure number 4. After that, we are going to examine our specimen using the low power and medium power of the microscope. So we are going to start with low power. Now, using the low power, I'm able to see some epidermal cells. We are also going to try using the medium power. Now, using the medium power, I'm also able to observe the epidermal cells of the onion. So after that, we are going to do the same procedure. We are going to repeat the same step, but now today, instead of using distilled water, we are going to use dilute iodine solution. Why are we using dilute iodine solution? We are using it to stain the specimen. Why do we stain the specimen? We stain them to improve clarity. So we have other solutions we can use, and these are methylene blue iodine solution. We also have erosin, sulfate. Also, we have fast green, among others. So we will repeat the procedure. Procedure number 1, you cut your bulb onion into 4. That was our first procedure. We did cut it into 4 parts. The second procedure, we are going to remove one fleshy leaf from the part. We did that. Third procedure, we are going to peel a piece of epidermis in the living part. So we are going to use a pair of forceps to peel it. And then after that, we are going to place a drop of iodine to the slide to stain my specimen. I'm going to irrigate the slide with the iodine solution and allow the iodine to enter the specimen through the cover slip. I'll use a blotch paper to remove excess iodine. Then I will observe my specimen once again, which is stained. Now I'm able also to see the epidermal cells, but this time they are more clearer than the first time. Thank you.